Hello everyone, today we'll be talking about polypeptide synthesis and why it's a central dogma of molecular biology. Well, first of all, it is the biological production of peptides, which are chains of amino acids, and polypeptides are multiple peptide chains joined together to create a linear model. Polypeptide synthesis is an integral part of the body's survival structure, an operation of cells due to its product, protein. Proteins control different characteristics of every organism, from building tissues and muscles, hormone production, enzymes, immune function, and production of energy. Thus, what we are overall is because of a different type of protein expression. Without polypeptide synthesis, life would have been small, and our existence would have been very different from what it is now. The DNA of every organism is stored safely within their cells and cell nucleus so that it does not alter or become damaged, and as a result, put the organism at severe risks. The DNA is responsible for each individual's genetic code. It is vital that it is untouched and never exit the cell's nucleus. Polypeptide synthesis is the cellular process of using the DNA within the nucleus to create proteins. The process occurs in two steps: transcription within the nucleus and translation. So let's take a closer look at transcription and translation. Both of these steps, they have three stages in them: initiation, elongation, and termination. Within transcription, the first step is initiation. This occurs in the nucleus and begins when the DNA molecule unwinds itself from the helix form at the gene location. The gene location is a portion of the DNA. This is used as a template to create mRNA or messenger RNA. When the genetic information is transferred from the double helix of the DNA to a single strand, the gene unwinds due to an enzyme called RNA polymerase, which attaches to the start of the gene where an enzyme called the promoter is. Then it pulls the two complementary strands apart. The complementary strands are bonded by nitrogenous bases, which all have a sugar, phosphate, and base. These nitrogenous bases in DNA are represented as the letters A for adenine, C for cytosine, G for guanine, and T for thymine. When the strands are pulled apart by the RNA polymerase, one strand is used as a template and is known as the template for mRNA. The process of elongation begins when the RNA polymerase moves along the template strand and adds base pairs that complement the template strand. The base pairs of mRNA strand do not include the nucleotide T due to the lack of thymine, so the RNA replaces this with the nucleotide U, also known as uracil. The mRNA continues to elongate, and eventually the RNA polymerase will reach the terminator where the last stage of transcription occurs: termination. The enzyme polymerase will reach the terminator on the gene strand, and as a result, the enzyme will cease to transcript. After the actual process of transcription, edits are made to the mRNA strand. Parts of the strand are deemed to be unstable and are cut out of the strand. The unusable parts of mRNA strand are called introns. Now, what are introns, and why do our cells make them? Until recently, they were thought to be junk DNA that had no purpose. Recently, studies have proven that there is proof that introns have an actual purpose and are not there accidentally. Introns are used within the DNA to provide instruction on how to bundle into chromosomes and to be stored in the nucleus. They were first discovered in 1977 by Richard Roberts. He thought that they were filler sequences that served no purpose. But after years of research and technological advances, scientists have discovered that the introns, which make up 90% of our DNA, have a purpose. The introns, also called non-coding sequences, are spliced out of the strand, and exons, also known as coding sequences, join together to form the mRNA sequence. To form the mRNA structure. Once the introns are spliced, They form into a lariat loop, and then are broken down by the cell. Introns are a vital part of polypeptide synthesis. The splicing of the introns allow the strand to be clearer instructions for translation and completion of proteins. Once the splicing of mRNA strands is completed, the mRNA is now mature and completed strands of exons leave the nucleus into the cytoplasm, where the next stage of polypeptide synthesis occurs. Translation is the process in which mRNA is translated into amino acids, which fold up into a protein. The mRNA holds information for precise synthesis of protein, and each type of protein to be made from an mRNA strand is decided by the codon arrangements in the strand. The process of translation occurs in the ribosomes, which are located in the rough endoplasmic reticulum. The mRNA binds to the ribosome and passes through the ribosome as amino acids are made into a peptide chain. The amino acid and complementary anticodon for the mRNA codons is brought to the ribosome by tRNA, also known as transfer RNA. When the tRNA carries an anticodon of 3 bases at one end and a specific amino acid at the other end, the molecule works with the ribosome to transfer the correct amino acid for the inclusion in a sequence to form a polypeptide. The tRNA molecule carries the start codon, and once attached to that mRNA molecule, the ribosome begins a process of elongation by matching the anticodons to the mRNA sequence. The polypeptide chain keeps elongating until any of the 3 stop codons UAA, UAG, or UGA are detected. Once a ribosome encounters a stop codon, it triggers a series of events, releasing a polypeptide chain. Each amino acid is then spliced off by its tRNA carrier. Once a polypeptide chain has been created, the last stage of translation occurs: termination. The peptide chain is then processed within the cell, and it may join to other polypeptide chains. And then it is mature enough to fold upon itself to become an efficient protein. Thank you for listening.